Thanks, Brett. So yeah, I guess this will be a little different than what we've heard before. So, as Brett mentioned, I'm a scientist at the Center for Infectious Disease Research. We're a nonprofit research institute up in South Lake Union. We're focused on global health and infectious disease, primarily HIV, tuberculosis, and malaria. So that all sounds great, but what does it have to do with tech? Well, it's true that most people at the center do things like that or this, which mostly seems to involve mixing small amounts of liquid together and making science.
And does it ever blow up when you mix those things together?
Well, there are warning signs around. I haven't seen it, but there are also a growing number of people like me. So my PhD was actually in physics, not in any type of biology, and I work on a computer all day. They don't even give me a lab coat. So with the advances of biotech over the last couple of decades, we can measure more and more things. We're generating larger and larger data sets, and in order to extract useful information from these data sets requires sophisticated computational approaches, machine learning, data mining. So that's what I do, and tonight I'll take you through a quick example of that involving tuberculosis. So I think it's fair to say that most people in this country probably don't think or hear about TB very much anymore, but it's still a huge global health problem. There are 1.5 million deaths per year from TB, which makes it the second leading cause of death from infectious disease. And there are actually 2 billion people who are infected with TB. That's over a third of the world's population. And so the WHO has set goals to eradicate TB by 2050, but it's clear that if we want to approach those goals, we need to develop new and better vaccines, treatments, and diagnostics. We're working on all 3 of those, but tonight I'll tell you about a new type of diagnostic. So this is the progression of a TB infection. So it begins, of course, when people are exposed to the bacteria and become infected. Is there a laser? No. It turns out that actually most people, at least initially, can control a TB infection. So they maintain the bacteria in what's called a latent state. But any one of these 2 billion people who have a latent TB infection, at any time, they could become sick with TB disease. But most of them won't. So the overall lifetime risk of developing active disease from a latent infection is only about 10%. The problem is we have no way to predict which of the infected people are at risk of getting sick. If we could predict that, then we could give those people prophylactic treatment, hopefully prevent them from ever getting sick in the first place, which would allow them not only to not get sick and potentially die, but it would also prevent them from transmitting the infection to anybody else. So this could have a real impact on the TB epidemic. And that's the goal of this project. We're trying to develop a new method to predict which latently infected individuals will progress to active disease. So in order to address this goal, we're working with several data sets. One of them is from our collaborators in Cape Town, South Africa. So they enrolled over 6,000 high school students and followed them for 2 years. And these students were seen every 6 months, and they were clinically assessed for having any signs of TB disease. If they looked completely healthy, a blood sample was taken. And then over the course of the 2 years, most of the students remained healthy, but a handful of them, about 40, developed TB disease. So now we're in this very powerful position. We have a bunch of blood samples. They're all taken from people who look to be completely healthy, but we know that some of these people are going to get sick and some of them aren't. So the question we ask is, can we use these blood samples to construct a biomarker, a classifier, to predict who's going to get sick? So the measurement that we're using to do this is gene expression in the blood. So we're actually measuring the expression of every gene in the genome in everybody's blood. Turns out to be about 140,000 measurements. In order to make models from this, we apply some machine learning approaches that we've developed that are well suited to biological applications. We're lucky to have several fully independent validation cohorts from multiple sites across Africa. So we took the classifier that we constructed, made fully blind predictions on our validation sets, and it's actually working pretty well. We can predict who's going to get sick up to 1.5 to 2 years before they're ever diagnosed with disease. It's not working perfectly well. We wouldn't expect it to, but we're involved in the planning stages of a trial to assess whether it's working well enough to have a significant impact on the TB epidemic. So if you're interested in learning more about this project or anything else we do at CIDR, here's our information, and I'd be happy to take any questions. Well, not this classifier, but we're also working on malaria. We're looking at vaccine responses for malaria and predicting who's going to be protected or not. With TB, we're also looking at treatment, predicting who's going to fail treatment based on their blood at the time treatment starts, predicting who's going to relapse at the end of treatment. So the blood is a powerful tool to predict these things. As long as you have a clear binary separation of two groups, you can try to form a classifier from it. Yeah. Yeah, so HIV and TB co-infection is a huge problem. Everybody in the study doesn't have HIV, so these are all just TB. They're all people who look completely healthy. That's the great majority of people who have TB. Of the 2 billion in the world, most of them don't get sick. So we're just looking at the people who aren't on any unusual drug regimens or have any other known co-infections. We are working on some projects looking at HIV, TB, and co-infection and trying to see if we can predict those as well. So hopefully in the future. Great.
Ethan, I'm just curious, what made you go work for CID instead of somewhere else?
Yeah, so I switched from physics at the postdoc stage, and I'm glad I did. I liked the research I was doing in physics, but it was very abstract and very far removed from the real world. So what I find so appealing about this is I get to work on really interesting research problems that will hopefully actually impact people's lives, and so it's kind of perfect. Yeah. We have a very active program for students. It's called BioQuest. I would definitely encourage you to look us up. We do tours for high school students all the time, and yeah. Right now, it's all internal. We have a fairly big cluster. Our lab has about 6 dedicated machines, and then the institute's clusters, about 20 machines. But we've looked into using some of the cloud computing, Amazon, Google, Microsoft, so we might start doing more of that. There's issues with some of the data. It's proprietary, so we don't know what we could use at this point. But.
Thank you.
Yeah, thank you.